Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, as you know that we are carrying a series on plant physiology, and under this very series we have so far conducted numerous lectures. In our previous session we discussed on the topic nitrogen assimilation. So dear friends, we would be continuing the topic further. We are going to talk further on nitrogen assimilation today also, and for this we have again with us in our studios Dr. Ashish Kumar Nandi. Dr. Nandi is Associate Professor in Department of Life Sciences, or you can say the School of Life Sciences at JNU. So dear friends, let's take advantages from his experiences and try to understand more about nitrogen assimilation. Hello sir, welcome to the EDICET lecture. Though we have discussed in depth on this topic in our previous session, but I would like you to explain us more about nitrogen assimilation. So
Sure.
Over to you sir.
Okay, thank you very much. Yeah. So, as you said that it is a continuation one. In the previous class we have discussed that why nitrogen is so important and what are the different sources from which plant can acquire from the environment, and also different forms like gaseous form, ammonia, nitrate and so on. And today we will continue to that, and hopefully I will be able to finish this chapter today. And the one of the important factor which we have discussed in the previous class is why actually nitrogen is very important, and like we have told that it is there in the amino acids, in hormones, in vitamins, in pigments like chlorophyll and so on. So, it is very important. And also what we have discussed that how nitrogen is acquired, there are only two different forms which are absorbed by the plant. One is nitrate, shown in the left side, and other one is ammonia. So, the bacteria in combination with the plant can make ammonia, that is a symbiotic nitrogen fixation. So, in the previous class also I have told you this is the energy reaction for the nitrogen fixation. Like one molecule of nitrogen, when it converts to 2 molecules of ammonia, it uses 16 molecule of ATP and 8 reduction potential. That means for every molecule of ammonia you need 16 ATP, so or 80 ATP and 4 reduction potential. So, that is a huge amount of energy that is required for nitrogen fixation. Also I told you that this reaction can be carried out in outside the biological system, that is done by the fertilizer factories by using huge amount of energy at high temperature and high pressure reactions. And in environment, in natural environment, many microbes can do that interaction with the help of an enzyme complex called nitrogenase. So, this is the nitrogenase complex and structure I have shown you in the previous class also. And what I told you that there is a paradox in nitrogen assimilation. The paradox is nitrogen complex is highly sensitive to oxygen. It does not work when oxygen is there, but at the same time you need oxygen for generating energy. So, there must be a balance, must be a mechanism by which the microbes can bypass the oxygen environment to make ammonia. There are two ways which are done. One of the way is in cyanobacteria. In cyanobacteria, they do a photosynthesis where photosystem 2 is missing and only photosystem 1 is present. So, there is no oxygen evolution and they can get energy albeit at a lower level in heterocyst and can have nitrogen fixation. The one which you have started discussing about symbiosis. Symbiosis is the combination of two individuals for benefit of both, and in this case it is a symbiosis between plant and bacteria. So, plant being the host provides carbon, all other nutrients, and bacteria takes gaseous nitrogen, convert to ammonia and give it to the plant, and that is how symbiosis works. So, this is a symbiosome formation where bacteria enters into the host and that get engulfed in a form which is called symbiosome, and the bacteria grow inside, which are then called as bacteroid. So, this bacteroid, when they are growing in the nodule, they have a different kind of morphology. They do not divide much, but they are specialized for nitrogen assimilation, whereas the ones which grow free, they are called bacterium. They divide actively and are not able to fix nitrogen. So, these are the nodule structures which I have also shown you in this slide before, and some of the nodules they grow round, and once it is matured it stops dividing like a determinate organ, whereas some of them it keeps growing like the one shown in the up top. So, this is a kind of a nodule which does not stop after certain growth, it keeps on growing, and there are different organs are made by plant which should look like as if it is a plant's own organelle. If you look here, look at this one, number 1. Number 1 is nodule meristem. That means it is a nodule, but it gets a meristem which is like a plant meristem. Number 2 is the zone of infection thread. This is the zone where new cells are getting infected infected mostly. This is the zone of expanding infection thread, and this is the fourth is the position. This is the large central part of the nodule where most of the nitrogen fixation go on. That when this is a plant cell, picture of a plant cell, and this plant cell is now hosting symbiosomes. So, these are symbiosomes hosting symbiosomes, and now when this takes place, like when this symbiosis takes place, there are a lot of new genes which are expressed by bacteria as well as by the host nucleus. This plant nucleus encodes a series of genes that are expressed only during symbiosis, and these are used for transportation of carbon, transportation of nitrogen, and symbiosis followed by ammonia assimilation. Now, I will tell you that how this nodule help in oxygen homeostasis. The word homeostasis is used to refer in physiology as a buffering system. I mean by oxygen homeostasis is that when oxygen level goes up, system should reduce it, or when oxygen level goes down, system should incorporate more oxygen so that nitrogen fixation can go on properly. This picture is here, this is the root of the plant and this is the nodule. A small part of the nodule is shown enlarged here. So, if you look carefully, you will see that this is the outer layer, or what you say is epidermal layer. Then these are the layer of cells which are similar to epidermal cells. This is the cortical cells, continues until you get the vascular bundle. Now, these cells over here are holding symbiosomes. So, they are fixing nitrogen. So, when I speak about the oxygen homeostasis, now there are many components which help in oxygen homeostasis. These are marked here, like here it is written as oxygen permeability barrier. This barrier, as it is shown over here, is covered throughout the nodule. So, there is a layer or there is a layer of cells which are surrounding the nucleus, and that cells have a barrier for oxygen permeability, or other words you can say selective barrier. So, it is not completely blocked. When oxygen is required, it allows to go in, but when oxygen is sufficient, it prevents the getting in. So, I will tell you now how this permeability barrier works. Other than that permeability barrier, you see the cells are also having leg hemoglobin that helps, and there is a cytochrome oxidase. So, I will speak about all of them. So, to begin with oxygen permeability, so and it would be interesting to know how cells can selectively allow oxygen to flow. So, to make you understand, I have drawn these figures over here, say possible mechanism of oxygen permeability. Just imagine that this is a cortical cell or the cell of the nodule which have the symbiotic bacteria. Say oxygen level is high. When oxygen is high, plants will be able to respire very actively, and naturally it will make lot of ATP. So, oxygen is high, ATP is high, and this high ATP level activates a potassium channel. Now, you see there are many types of potassium, there are potassium channels, mostly two types. One is called outwardly rectifying channel, another one is called inwardly rectifying channels. There is a outwardly rectifying potassium channel that gets activated with the high level of ATP. So, when potassium levels goes high in the cells, then what will happen, that this potassium ion will increase in the cortical cells, and when that happens, suppose this is the cortical cells. These cortical cells, now what will happen, when there is increased of potassium will lead to increase in the water in the intercellular space. So, when potassiums are pumped outside, see here, this is a potassium pump, this potassium will go out, and when the potassium goes out, the ionic concentration in the apoplast goes high. Say these are the, for example, these are the cells. So, when these cells will throw potassium outside, say these cells, this is the cells and this is air space. So, oxygen or air can flow as a gas between the cells. However, oxygen solubility is quite low in water. So, when this space is filled with water, then oxygen cannot flow much, and this filling of water is carried out by the potassium ion concentration, and this mechanism is somewhat similar to the way guard cells operate. So, the guard cells, when they are need to close, they accumulate more potassium, that increases the water content and that becomes turgid. So, here similarly, when potassium level goes high, it absorbs water. Now, what happened, like under low oxygen concentration. So, oxygen ion concentration is low, then ATP level goes down, and that will turn off the potassium channel. So, when potassium channel is turned off, then the potassium which was outside will now go in passively inside, and then that will restore the balance. So, when it restores the balance, then the water which was there in the between the cells will go off, and then the water space will open up. So, this is how the intercellular space in the nodule is controlled by water content, and then thereby control the flow of oxygen. So, this is a beautiful mechanism which is present in the nodule that allows oxygen to enter only when it is required. Now, coming to the next point, that is role of leg hemoglobins in oxygen homeostasis. So, this is also there in most of the books. However, this oxygen permeability barrier mechanism may not be there in many books. So, one has to read some advanced book like plant molecular biology and biotechnology kind of book that maybe I will be able to refer you. Here is the source of the book, which is Biochemistry and Molecular Biology of Plants by Buchanan, Grisham and Jones. A few more books are there. This leg hemoglobin is hemoglobin, all of you know, is a protein present in our blood that binds with the oxygen. So, when it binds with the oxygen, it allows more amount of oxygen to get dissolved in a liquid. So, having hemoglobin is actually increases solubility of oxygen in the water, or when I am speaking about blood, it increases the oxygen amount in the blood through hemoglobin, which can bind and allows to dissolve. This hemoglobin, and probably most of you would know that in our human body there are 4 proteins which come together as a homotetrameric protein, and that forms the complete hemoglobin structure. However, if you look at a similar oxygen binding protein that is present in our muscle cells are called myoglobin. This myoglobin is also oxygen binding protein, but that is a single subunit protein. The leg hemoglobin, which is present in the nodules, are very close to myoglobin structure. So, it is a single protein unit, and it binds with the oxygen, structure similar to myoglobin, and since this kind of protein was first identified in the legumes, they have been referred as leg hemoglobins. This leg hemoglobins can bind with oxygen, I told you. So, these leg hemoglobins are present in large numbers in cells which are undergoing symbiosis. So, every nodules cells are dumped with large amount of leg hemoglobin, and that leg hemoglobin can bind with the oxygen. So, now many of you may not know it, binding with the hemoglobin and oxygen, for example, or any 2 molecules when they bind together, they work in a kinetics. So, kinetics is like this. So, if A and B join together make AB, so how much of AB will be there will depend on how much of A, how much of B and how much of AB together. So, if these are kind of reactions are reversible reactions. So, if any time any of the amount A or B falls off, then the AB will break down to make individual component like A and B. So, similarly, you can see that suppose in a whole cell there are lot of hemoglobins and the peripheral area are exposed to the oxygen. So, in a cell when the peripheral area is getting oxygen, leg hemoglobin in the periphery of the cells will bind with the oxygen. So, what happened? The free oxygen content in the cytoplasm gets reduced. Now, these hemoglobin molecules or say leg hemoglobin molecules in the cytoplasm are diffusible. So, they diffuse, they move throughout the liquid and go on, and they will release oxygen only in the places where local oxygen is very low. The places which consume oxygen will have a reduced oxygen level. For example, mitochondria. Mitochondria is a place where oxygen is consumed. So, wherever the plant mitochondria is there, their oxygen is consumed. So, local oxygen gradient near mitochondria will be low. So, leg hemoglobin will release oxygen there. Similarly, the leg hemoglobin surrounding the symbiosome also will release oxygen. Then what will happen? That hemoglobin will be free, and again in the diffusion it can go to the periphery, bind with another molecule of oxygen and come down. So, essentially leg hemoglobin is binding with oxygen, increasing oxygen solubility inside the cell. At the same time, it is releasing the oxygen in a requirement dependent manner. The subcellular component where oxygen requirement is more, it will release there, like mitochondria and symbiosome, and the areas where it is not required, it will not release. So, in that way leg hemoglobins play very important role in oxygen homeostasis of the nodules. Now, that also we have to see what is the requirement level of oxygen between mitochondria of the plant, again bacterial respiration. You see the oxygen requirement by mitochondria and oxygen requirement of by bacteria are very similar. Both of them require for oxygenating the food particles essentially in the process of respiration, and both of them use in a system which is called as electron transport chain. The electron transport chain of plant present in the mitochondria is also bacterial origin. So, the electron transport chain present in bacteria and mitochondria are very similar, but quite interestingly in the process of evolution they got a different type of enzymes with the different type of requirement of oxygen. The better scientific term is different level of oxygen affinity. So, then I will tell you actually what is the difference between the bacterial bacterial cytochrome C oxidase. Actually, I did not tell you about that. There is an enzyme which is called cytochrome oxidase. This cytochrome oxidase can bind with cytochrome and can oxidize, and then the process oxygen is reduced to water, and that is how oxygen is used by electron transport chain. Now, the plant mitochondria which is there has a very low affinity for oxygen. So, if you I have a chart over here that you can have a look into here. So, you see this slide. This slide tells you the electron transport chain, and this is bit complicated. We do not have to look into it. There are complex 1, complex 2, complex 3 and look at complex 4. This is the complex 4 that binds with the cytochrome C, takes oxygen and oxidize cytochrome or the water is formed by reducing oxygen. This cytochrome C, look at the affinity for cytochrome C oxidase for oxygen. Plant mitochondria, the affinity is 100 nanomolar, whereas in case of bacteria which is free living has 50 nanomolar, and the amount of symbiotic bacteroid is 8 nanomolar. The affinity is defined at the concentration of a substrate by which the reaction undergoes to a saturation level. So, now if you are talking about oxygen binding with cytochrome C undergoing to a saturation in case of plant by 100 nanomole bacteria, and the same symbiotic bacteria, same cell where bacteroid is there, this is a very high affinity. When values are low, affinity is called high. Because it is a very high affinity, even up to 8 nanomolar level it can go to the saturation, whereas it is a low affinity one which can be saturated at 100 nanomolar level. So, now again if you think about the bacteria, again if you can think about the symbiotic cell. The symbiotic cell has both plant bacteria, mitochondria and free living bacteria. So, the leg hemoglobin which are surrounding the plant mitochondria. So, maybe there are about 100 mitochondria in a cell. So, wherever the mitochondria is less, mitochondria is there, leg hemoglobin is releasing oxygen there. Now, that oxygen has to cross 2 more barriers for reaching bacteria. Like as I told you, the symbiosomes have, like look at the symbiosome structure here. See, this is the cytosol. The cytosol is filled with leg hemoglobin and it is bound with the oxygen. Now, this oxygen can directly reach to the plant mitochondria, but for reaching the bacteria, it has to cross the endosymbiotic membrane, then bacterial membrane. So, it has to cross at least one more layer of membrane layer to reach bacteroid. Now, say this is the cell which has 100 nanomolar oxygen, bacteria plant mitochondria is using them, say very little amount 10 nanomolar is going in, and whatever the amount is going in are rapidly trapped by very high affinity cytochrome C oxidase present in this bacteroid. So, you can easily imagine that this bacteroid now which have the nitrogenase enzyme can work in absolutely oxygen free environment, where oxygen level can go as low as 8 nanomolar. So, and also bacteroid cytochrome C oxidase work with a low efficiency because they do not need much of respiration to do, as many of the metabolites and the energy is provided by the host plant cell. So, that is how a way bacteria can work in an oxygen free environment almost without and having still have a lot of energy made in their ETC and can provide ATP for fixing nitrogen into ammonia. So, there are 3 components I told you. One component is the plasma barrier, oxygen permeability barrier, that allows only controlled oxygen flow inside, and the whatever the oxygen comes in are trapped by leg hemoglobin, and that leg hemoglobin also allows more oxygen to remain in the cell as a buffer and selectively released to the mitochondria, and to a small extent released in the bacteroid containing symbiosome, and that is trapped by cytochrome C oxidase. So, rest of the bacteria systems remains almost an anaerobic environment where nitrogenase can do the function, and that is how it is work. Now, again bacteria have a 4th level of safeguard. That 4th level of safeguard functions in a gene regulation manner. Like many of you, if you are reading microbiology for example, or genetics or molecular biology, you might have come across with the word called signal transduction. The signal transduction is the process by which one external signal can be transported, and that can reflect to the physiology, behavior, and in this particular case I am talking about gene expression. And in bacteria, there are many genes are regulated or expression is regulated as an operon. So, this operon concept may not be new to many of you, where a large number of genes are regulated simultaneously by similar mechanism. These are called operons. These nitrogen fixing genes present in bacteria are also under operons. They are called a NIF operon or nitrogen fixing operon or fix operon, which are also called as fixation requiring genes operon. The transcriptionally also these genes are regulated in such a way that these genes will be expressed only when oxygen level goes down below the critical level. So, any amount of oxygen which is above the critical level, which can damage nitrogenase, bacteria will not make the nitrogenase enzyme that time. So, how that type of regulation works, I will tell you in shortly with the help of this slide over here. So, many of you probably know that bacteria has a system of receptors. So, here this is the receptor system of bacteria. These receptors are present in every living cell, be it animal, plant, insect or any kind of cell. These receptors, if you are talking about higher eukaryotes like plants and animals, they have a receptors where it can receive signal outside, and usually the receptor is a single membrane protein. In contrast to plant and animal receptors, many bacteria have a receptor where receptor is split into two, and that is why it is called two component kinase system of bacteria. So, kinase is an enzyme that can phosphorylate a substrate. Phosphorylate means adding a phosphate bond. This is a typical structure of a two kinase receptor, component receptor in bacteria. If some of you want to read more, you can just type in Google as two component kinase system, you will get lot of information to read. And in brief, this enzyme, this is the receptor split into two, one part is attached to a membrane. So, this is called histidine kinase. So, or it is written here HK, that had a histidine which is phosphorylated. Suppose this is the receptor, it can bind with the ligand, and the ligand binding would activate this receptor, the histidine molecule will be phosphorylated. So, this is the first component, and then the second component is called response regulator or RR. This response regulator has a aspartate amino acid. So, this is also called as histidine aspartate kinase system, where the histidine kinase component can phosphorylate the second component, and that becomes an active molecule. So, this is, you can think about a replacement of the MAP kinase systems which is present in the higher animal and higher eukaryote. So, like MAP kinases which get activated by phosphorylation, this response regulator get activated by phosphorylation and can turn on the other genes. So, for example, here you look here, this is a protein called FixL. This FixL protein is present in the bacteria surface, the bacteroid surface, which is present in the symbiosome. This bacteroid surface can be phosphorylated and can get activated. So, this is the first one, and second one is FixJ. The FixJ get phosphorylated to FixJ phosphorylate, and when they get phosphorylate, it activates the NIF operon and fix operon. So, this NIF operon, fix operon has the genes which are required for nitrogen fixation. Now, the catch is that the first step, the histidine kinase FixL will be phosphorylated only when oxygen level is very low. So, in other words, this is a protein which is present in the bacterial surface only. So, you can think about it, this is a system or antenna, this is an antenna for bacteria to read the level of oxygen. If the level of oxygen is low, then only it will turn on the nitrogenous genes, otherwise not. So, bacteria has a many different way, every system has many different ways how to protect themselves and be more importantly how to be more economical in terms of their resources. So far from yesterday's class and so far from today, I have told you how ammonia is made from gaseous nitrogen. But remember what I told you in the previous class is assimilation is only when a molecule or element is converting into organic form. So, conversion of nitrogen into ammonia is not assimilation. For assimilation, it must be incorporated into the organic molecule. So, this is what is referred as assimilation. There are few molecules which can receive ammonia. The most important reaction is carried out by glutamate. Many of you may be aware of glutamate. This glutamate is again an amino acid which is produced by carbon metabolism from alpha ketoglutarate or 2-oxyglutarate. So, this alpha ketoglutarate can convert into glutamate, and this glutamate, so alpha ketoglutarate or 2-oxyglutarate is an intermediate of carbon metabolism, and this can convert into glutamate, and this glutamate can take ammonia with the help of ATP. Again there is an enzyme called glutamine synthetase or GS. This glutamine synthetase can make it to glutamine. You see, this is an amino acid where NH2 COH and there is another COH end. So, this COH end or acid end at the tip of the glutamate can receive ammonia. So, you get a double amino group carbon component, it is a 5 carbon component which is called glutamine. This glutamine is the mostly as a first assimilated nitrogen in plant. So, this glutamine when it comes made, this glutamine can again react with another alpha ketoglutarate and can form 2 molecules of glutamate. So, this cycle is referred as GS-GO GATT, glutamine synthetase, glutamine oxyglutarate, glutamate. So, this is how the cycle continues. So, this is again the same cycle which is relatively easier way it is written, like say glutamine, glutamate is the recipient, it converts into glutamine. This glutamine can convert back to glutamate with the help of asparagine to aspartate, or it can form with glutarate to again glutamate, and this is how the nitrogen which is converted into ammonia gets into organic molecule. So, one important thing also I would like to mention over here, that for assimilation of any element, it is never that 2 elemental molecules combine to form an organic molecule. Assimilation always takes place by adding an elemental component into already present organic molecule. So, here glutamate is the organic recipient and ammonia is the inorganic recipient, that is from the organic molecule glutamine, and that is how assimilation takes place for any element. So, this is a Malate-Aspertate cycle which essentially I am showing you to show that there are many different, not only this oxoglutarate, there are many other molecules which take part in the active transport between symbiotic nodules and plant matrix, plant cytosol to convert nitrogen molecules. So, maybe this is Malate-Aspertate shuttle. If you want, some of you can read it from books, biochemistry books. So, this is how the schematics of symbiotic exchange, like you can see here, this is the glutamine synthetase. So, it exports glutamine, it can import photosynthetes. So, photosynthetes would come from the bacteria, from the plant, and then it would export the glutamine, and the cycle continues over here, which I have discussed some times back. So, this is the summary of nitrogen assimilation, whatever I have told you. So, this is essentially if you ask me to summarize and this ammonia assimilation, I would like to say that atmosphere has more than 80% of nitrogen, which is not used by any living being, and it is a proprietary enzyme present in the prokaryotes called nitrogenase that can convert nitrogen into ammonia, and this conversion is facilitated by symbiotic nitrogen fixation between plant and microbes, and this is carried out by an enzyme called nitrogenase, which is very sensitive to oxygen. This has two parts, one is dinitrogenase, another one is dinitrogenase reductase, and strategically placed iron sulfur, iron molybdenum sulfur clusters. This is what I have showed you in details in the previous class and told you how this helps to reduce nitrogen with the help of ATP. And heterocyst and symbiotic nodules help in providing oxygen free atmosphere for nitrogen fixation, and ammonia is primarily assimilated by glutamate. So, so far this is what about the nitrogen ammonia assimilation. Now, I told you that plant can absorb two different forms of nitrogen. One is ammonia, the other form by which plants assimilate is nitrate. Nitrate is quite abundantly present in the soil, and that if not, like in the agricultural land, the farmers apply large amount of nitrate fertilizers. So, nitrate is another source through which plants absorb nitrogen. This nitrate plants absorb, and if it is required, it would go to the plastate, where it would convert into again ammonia. So, plants cannot use nitrogen without making to ammonia. So, this nitrate would convert into nitrite, NO2 minus, this is NO3 minus, nitrate converts to nitrite. This nitrite goes into the plastate, where nitrite converts into ammonia, and again it will get to glutamine, which I just now told you by GS GO-GAT cycle. Now, if this nitrate is not used, not required by the plant immediately, it will go to the vacuole and it will be stored in the vacuole until it is required, then it will come out again. So, this is the simpler steps for nitrogen assimilation, nitrate assimilation. This nitrate, there is an enzyme which is called nitrate reductase, or in short it called NR, that converts into nitrite. This nitrite converts into ammonia by nitrite reductase. So, this is a two enzyme reaction process. This nitrate to nitrite is a cytosolic reaction. So, that means it takes place in the cytosol, whereas this nitrite to ammonia is a plastidic reaction. So, I will also tell you why this kind of different subcellular localization and subcellular requirement. The answer lies over here. If you look into the requirement, here nitrate to nitrite is one reduction step. It requires only one electron to convert. So, if you have a NADPH, it can form into NADP and then it can do the reaction. Whereas, nitrite to ammonia requires 6 reduction potential. So, it reduces 6 reduced ferredoxin and that 6 reduced ferredoxin converts into oxidized ferredoxin and the energy is used for ammonia assimilation. So, if you look, you will come to know that this is a very less energy requiring process, whereas this is a highly energy requiring process. Now, plants, if you look at total energy generation places in plant, there are mostly two. One is mitochondria and another is plastids. So, many of the plants green tissues are capable of generating energy through photosynthesis. So, by photosynthesis it requires gets energy of sun and that is converted through electron transport chain into ferredoxin and to ATP. And other place for generation of energy is mitochondria. In mitochondria, the food is used for generating ATP. So, theoretically, the reactions which are a plant can do it are in the mitochondria. Like if a reaction is highly energy demanding step, it is more favorable in the places where energy is more. So, there are two places such there, one is mitochondria, the other is plastid. However, mitochondria has a dedicated functions on respiration and there are very less number of metabolic reactions goes on in the mitochondria in contrast to the plastids. Plastids have, plants have very selectively are co-evolving along with the plastids by keeping the plastid genomes, large part of plastid genomes active, large number of genes are transcribed from the plastids and many of the bacterial metabolic processes, biochemical processes are still intact in say last more than 300 million years of evolution in the plant. So, since the plastids can generate energy, at the same time it can do a lot of biochemical reactions, many reactions in the plant that requires high energy are carried out in the plastid. For example, here what I am telling you is nitrogen or nitrate assimilation. Nitrogen to nitrite conversion requires single energy cytosolic and quite importantly again I am telling you here this nitrite is very toxic. So, any plant cannot accumulate nitrite to a great extent and that would be really very lethal and very toxic. And this nitrite, so whatever the amount of nitrite is made is immediately transported to the plastid and the plastid converts. So, the plants actually evolved in such a way that for the conversion of a toxic substance like nitrite, the energy should not be a limiting factor. So, it is transported to the plastid. In the plastid there is a plenty of energy. If it is a photosynthetic, it have a photosystems are there. In the photosystem 1 you get reduced ferredoxin made, that reduced ferredoxin can reduce nitrite and if it is a non-photosynthetic plastids there is a oxidative pentose phosphate pathway. So, some of you can read in biochemistry book about this oxidative pentose phosphate pathway through which a non-photosynthetic plastids can generate NADPH and reduced ferredoxin. So, this and that is why this toxic nitrite is immediately sequestered, you can say, in plastid, so that it can be readily converted into ammonia. So, now we also would discuss in details, and probably the time will be short here today, maybe in the next class, that how plant really keep that nitrate toxicity under control. And there are many levels of regulation by which level of nitrate reductase. So, I told you about two different enzymes. First enzyme is NR, nitrate reductase. Second enzyme is NIR, or nitrate reductase. Now, what I told you is nitrate is less toxic, though at a very high concentration it can be toxic, is relatively less toxic, but nitrite is very toxic. So, naturally, if you think a nitrate reductase or a nitrite has to be very low, then there are two enzymes. The enzyme which is making nitrite must be under tight control, and the one which is metabolizing nitrite must be very abundant, and that is what actually happens in the plant. In plant, nitrate reductase is abundant. So, whenever nitrate is made, that converts to ammonia, which is less toxic. However, the enzyme NR or nitrate reductase is under tight control, gene regulation. So, one of the regulation level is at a transcriptional level. Again, actually I have told you in previous classes also, and many of you probably know that genes are coded in DNA. This DNA is converted into mRNA, which is called as transcription. Through the process of transcription, a gene starts expressing, making mRNA, and that has to be again converted into a protein, which you call as a translation. Now, this nitrate reductase enzyme, which this transcription is light regulated. That means, when light comes, then only the nitrate reductase gene is expressed. So, that is, and again I told you that light is a major source of energy for photosynthetic plants. So, look at the way how plants keep a balance of this nitrate metabolism with carbon metabolism. Like at the transcription of an enzyme itself is regulated in such a way that it will be expressed only when there is enough energy there in the plant. So, it is not only for nitrate reductase, you will be amazed to know almost 1/3 of genes of a plant is light regulated. That means, out of every 3 genes, 1 is induced by light, and on an average, if you think about there are 30,000 genes are present in a plant, almost 10,000 genes are turned on at the very morning sun rays. So, whenever the morning first rays of sun reaches the earth, the plants express their 10,000 genes, which is almost 1/3 of the genes, and nitrate reductase is a part of it. So, that is highly balanced between nitrogen metabolism and carbon metabolism, and so that the energy is there, so that sufficient amount of energy is there for converting nitrogen into ammonia. So, in my next class I will tell you more about how this regulation takes place in details, and if possible some part of other elements how they are assimilated in the plant. Thank you very much for listening today.
Thank you, sir. Thank you so very much. As Dr. Nandi himself said that we would be discussing more on the other elements also which are required during the assimilation process. Dear friends, if you have any queries or if you want to give your feedback regarding this particular lecture, we would love to have your feedbacks as well as questions. For this, you can mail us at info.cc@nic.in. Dear friends, we would like to tell you all that we daily upload our lectures on the YouTube, so that you can access these lectures So for your convenience, for you to study your course material, I hope that these lectures are beneficial. So you can access today's lecture also. The lecture was on nitrogen assimilation, and the YouTube has its first part also. If you want to access it, you can definitely. Thank you, sir. Thank you so very much for giving your precious time. Thank you.